Assalamu alaikum. Welcome to my YouTube channel. Last time we have studied about the meiosis, and now today our topic is mechanism of enzyme action. As we know, when the enzyme attached with the substrate, a temporary enzyme substrate complex, it made the temporary compounds. Enzyme catalyze the reaction and substrate is the transformed into the product. It's mean the enzyme act upon the substrates and substrate convert into the products. After the after the enzyme and substrate complex the breaks the enzymes and the product are released. It means when enzyme act upon on the substrate is and form the enzyme and substrate complex and product is formed, then energy is released. We read about the enzyme and substrate complex. On this topic we have the two scientists have the two views and two different models. First model is the lock and key model. As we read in the order to explain the mechanism of enzyme action, a German chemist Emil Fischer in 1894 proposed the lock and key model. According to this model, the both enzyme and the substrate possess the specific shapes like this. Enzyme has the specific shape and the substrate has specific shape that fit exactly into another. In this picture, we see that both are exactly fit into each other. This model explains the enzyme specificity. Now, after the substrate and enzyme complex, the product is break down and convert the into the energy. The second model is the induced fit model of the enzyme. In this model, in the 1958, an American biologist, Daniel Kosheldon, suggested a modification to lock and key model and proposed the induced fit model. According to this model, active site is active site is not the rigid structure. Rather, it is molded into the required shape to form its functions. In induced fit model is more acceptable than the lock and key model of enzyme actions. As we see in the diagram in the figure 6.6, that enzyme and substrate are not specific shape, they molded into their required shape. So the induced fit model of enzyme is complete. Eh, It is it is stated that the Daniel Koshland model, induced fit model, is better than the lock and key model. And the other topic of enzyme is the specificity of enzyme. There are over the 2000 now enzymes, each of which is involved in the one specific chemical reaction. Enzymes are also substrate specific. The enzyme protease, which breaks the pep- peptides bonds in a protein, will not work on the starch, which is broken down by the enzymes amylase. Similarly, lipase enzyme act only on the lipids and digest them into the fatty acids and the glycerols. Specificity of the different enzyme is determined by the shapes of their active sites. Active site possess the specific geometrical shapes that it with the specific of substrates. In the specificity of enzyme, as we read that the enzyme and the substrate has the specific shape, and we see in the figure number 6.7 how to geometric shape of the active site of the enzymes determine the specificity for the substrates. Point out which substrate can exactly fit into the active site. As we see that the substrates 1 are not fit in the active sites and the substrates 2 are also not fit for the active site and the substrate 3 is fit for the active site. So this out of the three model, one model is fit in the active site. So this show the specificity of the enzyme. Now our topic of the specificities of enzyme is completed.